My name is Noelle Joyner, and it is September 7th. I am going to execute the DNA extraction lab. For the sake of time, I have gone ahead and put my peas in water already to soak overnight so that they're soft. And then I've also gone ahead and crushed them with the pestle. And then I added this salt as well. And after that, I'm going to go ahead and time jump and show me pouring the liquid into the test tube. I went ahead and strained out the solid with a cheesecloth, and I'm going to go ahead and transfer it to the test tube. Okay. After putting my solution into the test tube, I'm going to go ahead and add half of the detergent into the test tube. I'm going to go ahead and leave the solution in the refrigerator for 10 minutes, and then after 10 minutes have passed, I'm going to go ahead and get it again. 10 minutes have passed, and I'm going to go ahead and grab my solution and then add meat tenderizer. My solution has been sitting for 10 minutes in the fridge, so I'm going to go ahead and stir the solution. And then I'm going to add my meat tenderizer. Going to carefully stir the solution to not break up the DNA. And then I have my cold 91% alcohol, and I'm going to add it to my solution. And then I'm going to allow the contents to settle for 1 minute, and I will be right back. I have allowed the contents to settle, and I will go ahead and put up a picture of a closer look at my final product. What is deoxyribonucleic acid, and what is its role in living organisms? DNA is a polymer composed of two polynucleotide chains that coil to each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA contains the instructions needed for an organism to develop, survive, and reproduce. Why do we add salt in the lab? DNA's sugar phosphate backbone is charged. So by adding the salt, it neutralizes the DNA charge and makes the molecule less hydrophilic, so it's less soluble in water. The salt also helps to remove proteins that are bound to the DNA and keep proteins dissolved in the water. We add detergent in the lab for DNA extraction because the detergent is used to lyse the cell so that DNA is released into the solution, and then alcohol is added to cause the DNA to precipitate. Why do we add the papain powder? Adding meat tenderizer releases papain, which is a protease enzyme that breaks down the histone proteins and frees the DNA. Papain also denatures or breaks down the DNA that is present. And the purpose of chilled alcohol, if you use ice cold water and ice cold alcohol, it will increase the yield of the DNA. The cold water protects the DNA by slowing down the enzymes that can break it apart, and the cold alcohol helps DNA precipitate and solidify more quickly. In the end, my results of the DNA extraction, there was a good amount of yield. It looked like white stringy precipitate on the top of the tube, and then at the bottom was the green peas. And in the middle, my DNA did turn into a white precipitate at the bottom of the test tube, and then it moved up to the top and became white stringy precipitate.